the offsprings are not the same character as such in parental character. There is some variations. The variations occur due to the process of recombination, and this is called as crossing over. This is in naturally occurs in reproductive cells during meiosis division in plants and animals, any organism. But we can get the new character. That means new character means desirable character. In natural phenomenon, during crossing over or DNA recombination, maybe the next generation characters may be desirable or undesirable. But if we want to only desirable character, go through this technology, recombination technology. Let's see the steps in recombination. I think the brief introduction about rDNA technology is clear. So rDNA recombination, combination of two different genes. The combination to two different genes to get a desirable character in offspring, coming offspring. So that is called as rDNA technology. Due to rDNA technology, we get more desirable characters, right? Let's see the process or technique steps in rDNA technology. Right, steps in rDNA technology. For selection and isolation of desired gene, that is the first one. Second, selection of suitable cloning vector. Third, introduction of desired DNA into vector to form rDNA molecule. Fourth, rDNA molecule is introduced into suitable host. Selection of transformed host cell. Expression and multiplication of desired DNA in the host. Let's discuss one by one. First of all, right, selection and isolation of desired gene, right? Have you selected the desired gene? For example, this is one plant A. B. The same plant, even it is the same, for example, same mango plant, but among the different type of mango tree, A plant have high desirable character in the form of, in terms of high resistancy power, high yielding power. This character only comprising in this A only, but not B. Do you want to get only A type of character? First you selected the A plant, from the A plant, the DNA separated from the A plant cell. DNA separated from the A plant cell by enzymatic method. Later, we will discuss later the procedure of the how to separated the DNA, right? So you selected the first one of the desired character in plant. After that, from this plant, you separated a you isolate the desired gene. That's the first step. Next, after isolated from the desired character plant, you select a suitable cloning vector, because this desirable gene should be multiply in copy. Where it is multiply in copy, where the character shows easily self-replicable, those shows this type of character, this type of vector, act as a vector. So that first we select the suitable cloning vector, most probably vector from the bacteria, because the bacteria have the plasmid. Inside the bacteria have the plasmid. The character have the habit of self-replicating, and it is easily multiply in number. That's why I selected bacteria plasmid as the vector. This is the plasmid of bacteria because it can self-replicate easily. So selection of suitable vector also. Desired gene, okay. Now the vector, okay, plasmid in the form of plasmid, different type of plasmid there. Any one plasmid selected as a suitable vector. Right. The second step also completed. First and second completed. First step DNA, second step vector. Here the vector is plasmid. Vector is plasmid. Not only plasmid, we can use the cosmid also. Bacteriophage also, different type of there. Next, introduction of desired DNA into vector to form rDNA molecule. To form rDNA molecule. Now, this desired DNA introduced into the vector plasmid to form rDNA. For example, here, so this is the DNA, desired DNA. For suppose the plasmid in the, let me change color. For example, plasmid in blue color. Plasmid in blue color. Right here. So cut the some place and the place inserted with desired DNA. As a result, it appear as a plasmid and desired DNA. Right? So what are present in this? In this desired DNA present and plasmid present. For that, first you cut the some portion of the plasmid and now later the desired DNA inserted into the plasmid. Now it is called as rDNA molecule. This is called rDNA recombination. Recombination occur. Recombination means already told you recombination DNA two different type of genes. What are the two different type of genes here? Here, just wait. Two different type of genes are one is mango plant desired DNA. Second one is plasmid. So plasmid genome different, plant genome different. That's why it is called as rDNA molecule. Right. Now rDNA formation clear. In the fourth step, rDNA molecule is introduced into suitable host. So what is our concept? For concept, we get the multiple copy of desired gene. Is this gene can multiple in the same plant? That means in plant, the plant it cannot multiply in rapid number. The only the desired gene, whatever it may be, any gene can multiple replication reformation formed within the plasmids only. That means suitable vector only, because the plasmid have the self-replication capability. That's why when we inserted this desired gene into plasmid, now it is get converted into rDNA. Now this rDNA need to be multiple for rapidly multiplication. Should get rapidly multiplication. So get rapidly multiplication, we supposed to introduced into the host. Suitable host means here actually where is the plasmid where we isolated, from where we isolated the plasmid, from we are isolated from the bacteria only. So the suitable vector should be bacteria. Now again this rDNA introduced into the suitable vector means suitable bacteria. Most probably Escherichia coli and different type of there. Now, when it is introduced, now the fourth class also. Next to selection of transformed host cell. Now this cell called as transformed cell. This cell called as transformed. This cell is called as transformed. This slide. Now this cell called as transformed cell. This cell called as transformed cell. It's called as transformed cell. What is transformed cell? What is normal cell? Normal bacterial cell, transformed cell. Normal bacterial cell only genome present, but transformed cell rDNA or recombinant genome present or recombinant DNA present. So that is the transformed host cell. Now selection of transformed host cell. Actually, after formation transformed cell, what is the characteristic nature of the bacteria? Bacteria, but during binary fission, it can multiple in number 2 to 4, 8, 16, 32. Finally, it forms the uncountable number of bacterial cells. In each and every bacterial cell, when it is divided, the rDNA also present. Here also rDNA, RDN, rDNA. Here also rDNA, here also rDNA. Now, like that, number of rDNAs are formed. Number of rDNA formed. But we should make sure whether the transformed host cells present or absent in the group of the cell. After formation, different type of group of cell, it may be some are not having rDNA, some are rDNA present, so that we can select the only transformed cells and ignore the rest of the cells. Selected only rDNA or transformed host cell. Next, expression and multiplication of desired DNA in the host. Later, when we introduced into the host cell, later this desired character, that means what character you get, what character you isolated, these iso character they express in the next generation plant. So let's see again in one by one. So this is the steps in rDNA technology. First, selection of desired gene missed from the host. Selection of suitable cloning vector means plasmid. Introduction of desired DNA into vector. As a result, formation of rDNA molecule. rDNA molecule is introduced into suitable host. Now, when this enter into the suitable host, it is called as transformed cells. Now, expression and multiplication desired DNA in the host. Finally, the desired character expressed in host cell. So these are the rDNS technology steps. Is it clear? Next one, let me take the slide. Right, as I said, first this. Selection and isolation of desired DNA insert. Right. In rDNA technology, the selection of DNA segment of interest which is to be cloned. Actually, already told you, first we selected the DNA segment from the desired character plant. The desired DNA segment isolated enzymatically with the help of some enzymes. For example, actually, I gave an example of desirable character mango tree. From the mango tree, we supposed to separate the cells. From the cells, we treated these cells treated with enzymes such as pectinase, cellulase. As a result, cell wall dissolved. Now the nucleus separated, and with help of nucleus enzyme also, the chromosome separated. When finally with help of some chemical cell centrifugation from the chromosomes DNA separated. The DNA fragment get obtained. Now this DNA segment, this is ready to clone. This DNA after separated, the DNA is ready to clone. Now let's see the methods of separation isolation of the desired DNA. First one, mechanical sharing. Second one, shotgun method. Third one, RNA directed synthesis. First, let's see mechanical sharing. This method involves fragmentation of cellular DNA. Fragmentation. Fragmentation, that means breaks into number of pieces of desired character of cellular DNA. by mechanical force. Due to the mechanical force, what will happen? The cell break, not only cell, the cellular DNA also break into number of pieces. Now, um, right. And also in this mechanical force using sonication ultrasound. This sonication ultrasound can cut the DNA into fragments about 300 by long. So DNA solution may be stirred at high speed. Later, after break into pieces, now centrifugation with the high resolution RPM value, the mainly 3000 to 30,000. As a result, due to this RPM value, the DNA are separated. Right. So anyway, we can isolate it with help of mechanical sharing, mechanical force in the form of sonication with ultrasound. That is one method. And shotgun method. In shotgun method involve the enzyme restriction, digestion, cellular DNA enzymes using specific restriction endonucleases. In this shotgun method, using restriction endonucleases, when we treated with endonucleases enzyme, where it is, just wait. Endonuclease, endonuclease enzyme, we can get the variable DNA fragment. Right? These DNA fragments are cloned into suitable vector. That is the next step. So in every method, whether it is mechanical method, shotgun method, or DNA method, after we get the different type of DNA fragments, this inserted into the suitable vector. Why we introduced into suitable vector to get rDNA, which is next it is transformed into recipient bacterial colonies. These are called transformed cells. Thus, each and every DNA fragment of cellular DNA is cloned in the form of bacterial colonies. Later, the bacterial colonies are formed. But this actually, first what we get the number of DNA strands, number of DNA strands 2, 3, 4, but we don't know where the desired DNA present, where the desired DNA present. So, do you want to get this desired DNA? First of all, these all broken DNA or broken genomes introduced into the vector. After vector, it is transformed into the host. After formation transforming cells, you selected the where the desired DNA present with help of probes, with help of radioactivate probes. Right. In the next method, RNA directed synthesis. In RNA directed synthesis, in reverse process, for example, this is the one mango plant desired gene, not only gene, for example, mango plant DNA. From this DNA strand, in reverse manner, from the DNA, we get mRNA. We get mRNA. This process called as reverse transcription. Reverse transcription with enzyme involved by transcriptase. Transcription. It was discovered by Tamin and Baltimore. Tamin and Baltimore. Tamin and Baltimore. Right? So that what happened actually in this process, first desired DNA, from the DNA reverse process mRNA prepared, not DNA, mRNA prepared. From the mRNA we can, during translation process, we get protein strand. We get protein strand also. It is another different species, another scenario, but after formation mRNA, this mRNA to be cloned, right? These are the first step. Next one. After next step, what is the next step? Selection and isolation. Selection and isolation of desired gene. Next step is selection of suitable vector. Let's see suitable vector here. Selection of suitable vector, right? Right. Selection of suitable cloning vector. Already told you the cloning vector is self-replicating. It should be self-replicating DNA molecule. It means the desired DNA is to be integrated. A suitable cloning vector is selected in the next step for the rDNA technology. Most of the common use vector is plasmid. I already told you the plasmids are most appropriate vectors and bacteriophages and also cosmids. Cosmids also we can use. Next, introduction of desired DNA into vector to form rDNA molecule. Third step, introduction of desired DNA. You get desired DNA from the host, from the any host, that means mostly plasmid present in bacteria. First of all, bacteria, plasmid, another genome. When we treat it with some chemicals, these bacteria such as sodium hypochlorite, some detergents, the cell wall of the bacteria dissolved. Now we easily get separated the plasmid. Next, introduction of desired DNA into vector to form rDNA. In first step, we get desired DNA in by method of mechanical or shotgun or reverse transcriptase manner. So anyway, in any method, any form of method, we get our desired DNA, and in second step, we get the cloning vector in the form of plasmid. Now we need to integrate it both desired DNA inserted integrated with the vector. That means DNA vector. Now it is called as rDNA. Called as rDNA. Now it is called as rDNA. Right? Right. So actually, here this is desired DNA, this is plasmid. But to joining this both DNA with help of the ligase enzyme. Ligase. With help of ligase. Both the plasmid DNA and desired DNA both are fused. Now this is called as rDNA. In next step, rDNA introduced into the suitable host. Already told you host is the bacteria. Let me take a slide. Right. Selection of suitable cloning vector. Introduction of rDNA. Okay, this is also completed. rDNA molecule. After get rDNA, I already told you how the formation rDNA. rDNA plasmid genome with desired genome. Right. After formation rDNA molecule, it is introduced into the suitable host. Suitable host. Suitable cells are selected and the rDNA molecule so formed is introduced into the host cells. This process entry. What happened in this fourth stage? Entry of our recombinant DNA into the host cell. Already told you host cell where the existed plasmid that is act as a host cell. So actually we isolated the plasmid from the bacteria. Again we injected into the bacteria. So after introduced into bacteria cell, now the transformed cells called as transformation This process called as transformation. Usually selected hosts are bacteria cells like E. coli, yeast, also fungi may also be utilized. So these are all act as a suitable vector because if we take E. coli bacteria or fungi, these are all can self-replicated and rapidly replicated within a short span of time, right? Next, selection of transformation cell or transformed cell. The transformed cells are that mean recombinant rDNA cells. rDNA cell that is called recombinant cells are those host cells which have taken up the rDNA molecule to be separated from the non-transformed cells by using various method making use of markers. We can use different, we will discuss about the marker genome markers later. With help of the genome markers from the group, already told you after formation, this is cell, this is cell, this is rDNA. After multiplication, the group of cells are formed. First division, second division, final difference, some divisions. Group of cells formed. But this group of cells may not all transformed cell. You with help of marker genes means the marker is complemented to the genome gene. Already radio labeling with this marker desired gene, the same sequence with help of the markers utilized for identify the transformed genes. If the markers utilized, this marker exactly suitable to the desired genes in the group. The rest of the considered as a non-desired genes. Now we separated non-desired cells, non-transformed cells from this group. Next, expression and multiplication desired gene. Finally, it is to be ensured that the foreign DNA inserted into vector DNA expressing the desired character in the host cell. And also the transformed host cells are multiplied, obtain sufficient number of copies. If needed, such genes may also be transformed to express another organism. So if this after formation transformed cells after get the desired character, when we introduce it into the another organism, in those organisms, this desired character expressed later. So this is rDNA steps. This is rDNA steps, right? It is look like a tissue culture techniques. But in tissue culture, in part cell direct or protoplast culturing. But here the culturing of say two different genomes, only genomes. So that is the concept of rDNA technology. Right, next one. Just wait 1 minute. Right, sorry for inconvenience. Okay, let's summarize once again rDNA technology. So rDNA technology, we get desired character from joining of two different genes. First, in this process, we selected the host. Sorry. selected the host cell from the host cell, separated. Where it is? Just wait. Right. So from host cell, we separated desired gene with help of enzymes. After, and also this separation isolation with help of mechanical method, shotgun method, and by reverse transformation method, reverse transcriptase method. So after get the DNA fragments, these DNA fragments get introduced into suitable vectors. If the suitable vector, where it is switched, right? Suitable vector may be plasmids, plasmids, or bacteriophages. We need to separate the plasmids and bacteriophages from the bacteria, plasmids from the bacteria. Same by enzymatic method or some chemicals also we can utilize sodium hypochlorite, especially sodium hypochlorite detergents. After separated plasmid from the bacteria cell, the plasmids and desired gene both get incorporated as a result formation of rDNA. After formation rDNA, this rDNA introduced into the suitable host. So vector we get in the form of plasmid, genes also we get, and after developed rDNA, this rDNA recombinant DNA introduced into the suitable host. After introduced into host, it is becoming a transformed cell. Due to the this cell division or multiplication, the number of group of cells are formed. Those are all cells, rDNA cells. So maybe in this group, some are not having rDNA. We need to separate those not having rDNA from this group. So finally, we selected. We introduced into this desired gene another organism. We get the desired character, right? So these are all the rDNA techniques. Let's see the diagrammatically. Just wait, where it is. Diagrammatical. Right. Just wait. Diagrammatical representation where it is. Technology. Yeah. Is it visible? Host, BRA host. Not showing here. Right here. I think now visible. . Right. So let's see diagrammatical representation. Here, for example, so this is the plant nutrient from agrobacterium. It is a bacteria. This normal genome, this is plasmid genome. We get separated plasmid, and also DNA contains gene of interest. This is also separated from the desired plant. That means high yielding, high resistance power plant. From high resistance power plant, we separated the gene. Also from the bacteria, separated the plasmid. And next steps. Now these two, that means the plasmid and also desired gene, both are incorporated. Now, as a result, it is called as recombinant DNA or recombinant DNA plasmid, TI plasmid, right? But with help of the exonuclease enzymes, nucleases enzymes, we will discuss later, nucleases or endonucleases enzyme, join with ligase enzyme. Ligase enzyme also we will discuss later. So anyway, now actually this again introduced into the host cell, another host cell not here not missing. Another host cell means another bacteria. Same introduced into bacteria. After get number of cells, we selected from these cells only desired gene cells only. Desired genes transforming cells selected. Now these desired genes transforming cells introduced into the another plant, new plant. Introduction methods also we will discuss later in further units. How the desired gene get incorporated into the new plant. We will discuss later. So after that, after we introduced into the plant, later this plant shows the some desirable character, right? So this is the rDNA technology. Next one. Some enzymes used in rDNA technology. So already told you some enzymes involved in rDNA technology for cutting, for joining, for those purpose. Though in this most important are restriction endonuclease enzymes, polymerase enzyme, ligase enzyme, nuclease enzyme. They are all most important. Let's discuss one by one. Restriction enzymes. Restriction enzyme cut the DNA fragment. The function of restriction enzyme cut the DNA fragments. So in this there are three types: type 1 restriction endonuclease enzyme, type 2 restriction endonuclease enzyme, and type 3. First, let's see the type 1 restriction endonuclease enzyme. The type 1 endonuclease enzyme the cleave only one strand of DNA. One strand of DNA. These enzymes have been recognized the sequence of 50 base pair length. Let me take the pointer. Right. So this for type 1 endonuclease enzyme cleave only one strand of DNA. One strand of DNA. Actually, DNA have the two strands, right? DNA have two strands, made up of two strands, but it is cleave separated only one strand. These enzymes have recognized the sequence about 15 base pair length. They require magnesium ions and ATP for their function. Without magnesium ion, without ATP, the restriction type 1 enzyme not work further. Such type of restriction endonuclease give the DNA about 1000 base pair away from the 5 end, not 52, away from the 5 end. So not 52, 5 end. 5 end. And 5 end, not a 2, 5 end of the TCA located within the recognition site. So here the recognition site, the recognition 15 base pair. For example, this is the recognition site. Recognition site After attach the DNA polymerase 1, this DNA polymerase 1 cleave the almost 1000 base pairs. This is 5 end, this is 3 end. This cleave away from the 5 end. This 5 end, 5 end to cleave site 1000 base pair present. So this is the type 1 function. Let's see type 2 restriction endonucleases. Actually, these type most important endonucleases for the gene cloning and RDNA technology. These enzymes are more stable. They show cleavage only at specific site, and therefore they produce the DNA fragments of defined length. These enzymes shows cleavage in both the strands. And cleavage not only one strand, cleave in both the strands. So that is restriction endonuclease 2. And next one, wait, just wait. Right. So type what the difference between type 1 and type 2? In the type 1 only cleave the one strand, but not two. But the second type, the enzyme show cleavage in both the strands of DNA. And they're requiring magnesium ions for their functioning, but don't require ATP for cleavage. They case cleavage both the strands of DNA. That is the difference. In type 1, require magnesium ion. Type 2 also magnesium ion. But only one difference is another difference is first different one cut only one strand, type 2 cut two strands, and second difference is in first type require ATP to cleave the DNA, but the second type don't require ATP for cleave the DNA. The type 2 restriction endonuclease are used to gene cloning or RDNA technology due to their suitability. That means it can the both stand, it can without ATP. For this stability, we can use more properly. The recognition sequence for the type 2 restriction endonuclease enzymes are in the form of palindromic sequence. For type 1 only the recognition 15 base pair with 15 base pair away from the 5 end. The distance between 15 base pair to 5 end, in between 1000 base pairs are present. But in second type, already told you it is a specific. It recognizes the specific sequence. That specific sequence called as palindromic sequence. This type of palindromic sequence not present in type 1. The palindromic sequence may be G, sorry, right, GAATTC, and in reverse manner GAATC. Just wait. GAATC can get showing here. So anyway, not shown here. So just remember the palindromic sequence GAATTC, and in reverse manner also GAATTC, parallel manner, anti-parallel manner. Now this is palindromic sequence. Only at the place of palindromic sequence, this type of two restriction endonuclease cut the DNA fragments. That is about DNA polymerase 2. And let's see sorry, right. It is, right. Let's see DNA polymerase 3. Right. Let me take the pointer. Type 3 restriction endonucleases. These are not used for gene cloning. Type 1 used in gene cloning. Type 2 used in gene cloning, but not type 3 not involved in gene cloning. They are the intermediate enzyme between the type 1 and type 2. They require magnesium ion like type 1, type 2, and ATP for cleavage like type 1 only. And I told you type 2 not required ATP. They cleave the DNA at a well-defined site in the immediately vicinity of the recognition sequence. That means here also like a type 2 recognition sequence present. After recognition sequence, the with help of the ATP type 2 cleave the DNA. Right? Example HINF3 etc. The nature of cleavage produced by restriction endonuclease is considerable importance. The cut the DNA molecule in two ways. One is blunt ends. Another one is restriction. Both strands DNA simply at the same point with the special sequence recognition sequence. As a result of this, the cleavage, the DNA fragment with blunt ends. That is first one. Second method is sticky ends. Second method is sticky ends. For example, blunt end means so this is the specific sequence. For example, this is specific sequence. In the specific sequence, we cut the DNA at the same equal site like this. As a result, it is separated into two of like this. Actually, not correct. This is double strand DNA. For example, this is one strand. This is one other strand. Now, cut with help of the three. As a result, make it off like this. Like this, CC would stand only, GG stand only. CC stand only, GG stand only. Is there any complementary to attach? Is there any complementary nucleotide to attachment? No, because they are the C and here G end. There is no complementary. That's these stands called as blunt ends. Now another one is sticky ends. In sticky ends, change the shape. Actually not given, just wait. For example, CCC. This is cut at one strand cut at this side. Another strand cut at this side. Another strand cut at this side. What would be now? It is formed as a C C strand. Next G G G C. Another separated is like this, for example. C G, 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 and CC. This is the lip change. In this manner, cut. There is a space to attachment. There is a space to attachment. For example, here CC only, rest of the empty. So there is a space, there is a gap to attach it with another DNA. Guanine, 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 cytosine. So the guanine cytosine attached with cytosine, cytosine, guanine. That means attached with cytosine, cytosine, guanine. From these nucleotides attached to this strand, these are called sticky ends. These are called sticky ends. So anyway, after cut into two actions, one is blunt end and sticky ends. After finally they get developed into rDNA. Final rDNA formed later. Another one is exonuclease enzyme. This exonuclease enzyme that remove the nucleotides. DNA, DNA, adenine, triple bond, thymine, thymine, adenine, triple bond, double bond, sorry, guanine, triple bond, cytosine, thymine, double bond, adenine. Like this, hydrogen bonds are present. With the help of this exonuclease enzyme, the activity of exonuclease enzyme first remove these nucleotides from the one end to another end. An exonuclease remove the nucleotide from the not 52, not 52, not 32, 5 end, 5 end like this, 3 end of DNA molecule. An exonuclease never produce internal cuts in DNA. Actually, restriction endonuclease enzyme cuts internally, but the exonuclease enzyme activity never cut internal, never cut the strand, just remove the nucleotides. As a result, the two DNA free, make it free. In RDN technology, various types of exonuclease enzymes are employed, like exonuclease ball 31, exonuclease 3, lambda exonuclease, such as. So this is the exonuclease activity. If we see ligase activity, I already told you the function of ligase. Ligase, the ligase enzyme to attachment to different genes. This ligase enzyme action opposite to the nuclease or exonuclease. What is the exonuclease? Exonuclease cleave the nucleotide. What is the function of restriction endonuclease? Cut the DNA, cut the gene at the site of in between the nucleotides, whether it is blunt cut or sticky cut. The ligase join 5 and phosphate to 3 and hydroxy group. Already we have discussed in cell biology topic. Phosphoric acid 5 and 3 and hydroxy group of these two nucleotide ends establish a phosphodiester bond between them. So with help of ligase enzyme, one of the gene 5 and phosphoric acid attached with another enzyme 3-hydroxy group with phosphodiester bond. Thus, they are capable of joining together two individual fragments of double-stranded DNA. The process is called ligation. The process called ligation. All living cells produce ligase, but the enzymes used in genetic engineering are usually purified from the E. coli. So these ligases also we get separated, isolated from E. coli bacteria that have been infected with T4 phages. The T4 phages enzyme also the function same like ligase. That means it joins the two fragments with 5-phosphate and 3-hydroxy. So this is the function of DNA ligase. Next one, polymerase enzymes. Polymerase enzyme, right? Polymerase enzyme. Polymerase enzymes, three types: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Right, time is over. We will discuss later about polymerase enzyme. Okay. Tomorrow we will discuss about polymerase enzyme. Thank you all.